Okay. Okay, so we have been talking about meiosis. You should have your notes out. And we have some questions to answer, but let's remind ourselves, first of all,
Thank you.
and you don't need to draw this.
Wouldn't that be nice to be able to do? Yeah, like guess when I get to go home tonight? Yeah. Guess when I'm gonna go home tonight? 8:30. Well, I don't even get to go home from school. Oh, that is so rude. It's so rude.
Are you creating a community? No.
Okay, so again, you don't need to draw this, but let's just remind ourselves of what the goal and what the difficulty to overcome here is. I've only drawn out 5 pairs of duplicated chromosomes of mom and dad. So there's mom's duplicated chromosome number 1, and there's dad's duplicated chromosome number 1, and here's mom's duplicated chromosome number 2 and dad's duplicated chromosome number 2. And our goal is we've got 4 chromosomes that are needing to be divided up into 4 different cells, right? I've got 4 chromosomes that are needing to be divided up into 4 different cells because I want 1 chromosome in each sperm cell or 1 chromosome in each egg cell. And this chromosome has to somehow magically find this chromosome, and this chromosome, has to somehow magically find this chromosome, and this chromosome has to somehow magically find this chromosome, and let's get that out of the way, and then this chromosome has to find this chromosome, and then this chromosome has to find this chromosome. And then they have to, in prophase of the first meiotic division. And what did we say the term was for chromosomes of mom and dad's duplicated chromosomes that have the same chromosome number are called? Starting with an H. Homologous. They're homologous, right? These are homologous chromosomes. They have to find each other. And then they have to form an association with each other. They have to get together and form a tetrad, right? And this happens in prophase. They have to form an association with each other. And what do they do when they form that association with each other? First of all, what was that association called? The group of 4 chromosomes together was called a what? Not tetrus, but a tetrad, right? It was a tetrad, right? The group of 4 chromosomes together was a tetrad. And what did they do while they were in that physical association? This should be a review for you guys. You should all know this. What happens in Prophase One of Meiosis? And how does it help accomplish the goal of what meiosis does? What do the 4 chromosomes do in Prophase 1? They what? They pair up, but I think I just heard somebody say that. If I have this chromosome and this chromosome in close association, what can they do? Aren't they going to switch genetic material? They're going to shuffle the genes, right? Mom's DNA is going to get switched with dad's DNA. And it's a random process, right? We don't switch the same things every time. We might switch these 2 pieces right here, and we might switch these 2 pieces right here. And they have to be exact matches, right? I have to cut out this piece of DNA from mom's chromosome and this piece of DNA from dad's chromosome, and they have to switch places, and they can't be off by even 1 base pair. And each set of homologous chromosomes is going to do it a bunch of times. They're going to switch that genetic material, and this is accomplishing the purpose of genetic variation, right? That's how we introduce genetic variation into a population. Sexual reproduction accomplishes that through this process. Otherwise, what would be the only way that I could introduce genetic variation? If we didn't have this kind of a process where the chromosomes got together and switched genetic material like this, what would be the only way to introduce genetic variation into a population? Yeah.
Having scientists do it?
Okay. Artificially or naturally, how would that occur, Saskia? Mutation, right? Mutation is going to be the only way that we get genetic variation in this. So in order for this kind of a thing to work, in order for population genetics to work, we have to have this sexual reproduction where we introduce the idea of switching genetic material between chromosomes like this. Okay? So that's really, really, really key to meiosis. That's part of the point of meiosis. And you have to understand that. Okay? Again, that whole idea should just be reviewed for you guys. You've all had this before. But anyway, this is Prophase 1, right? We get together, we form these tetrads, we switch genetic material, we shuffle the deck, and then we have a whole bunch of other problems. This whole interaction here where we're swapping genetic material is fraught with peril. If I mess up by even 1 base pair, I introduce fatal genetic flaws. And we said before, it's estimated that over 50% of conceptions never make it past the first few days because this is such a complex process and it can screw up in so many ways. Okay, now we have some questions here. And I want us to think about these. And hopefully these questions will help us solidify our understanding of this process. And I want to sort of block my answers so that you can't see them until I'm ready for you to see them, because I don't want to write this out a second time. Okay, why do you think each of the homologous chromosomes has to line up with its corresponding partner on the metaphase plate at metaphase? Why do I have to have it so that this chromosome 1 from mom and this chromosome 1 from dad has to line up on the metaphase plate together at this time? Yeah.
It ensures that the genetic material is evenly distributed.
Okay, that's probably an even better way of saying it than I said it. That's more concise. I said this ensures that one pair of each chromosome goes to each of the first two daughter cells. But I like the way you said it as well. But since I've already written it, I'm going to say let's write this down. But it ensures that, when, how did you say that again?
That the genetic material is evenly distributed.
Okay, that's the idea, right? We want one of these to go one way and one of these to go the other way. It ensures that the genetic material is evenly distributed. It ensures that one pair of these goes one way and one pair of these goes the other way. In the first meiotic division, the daughter cells each get one pair of chromosome 1s and one pair of chromosome 1s, one pair of chromosome 2s and one pair of chromosome 2s, one pair of chromosome 3s and one pair of chromosome 3s. Okay? That's why this has to happen. They have to line up with the corresponding partner on the metaphase plate at this time. Okay? You're seeing my answers here. And how is this accomplished? Oh, come on. Well, I've kind of blown it already, haven't I? So the homologous pairs are already attached, right? That physical association that they formed during Prophase 1 means that they're still attached together when they get to the metaphase plate. Okay, they don't break that association. They're still stuck together. That kind of makes it so that they have to be at the metaphase plate at the same time. Christian, are you using your computer to take notes, or?
Yeah.
Okay. Okay. Good. Does that make sense? They're still stuck together.
Can you read that again, Dr. Marshall? It's just kind of hard to see.
I'm sorry. The homologous pairs are still attached from the formation of the tetrad during Prophase 1.
Thank you.
The homologous pairs are still attached from the formation of the tetrad during Prophase 1. And I hope you agree that by understanding what the purpose of meiosis is, that helps you remember what happens. The purpose of Prophase 1 is to shuffle the deck. That means in order for me to do that, I have to form this association. So correlate the goal of meiosis, what is that goal of meiosis, with what you would expect to see. Okay, if I'm going to shuffle the deck, I have to form this tetrad, right? And then, oh yeah, by the way, they're still stuck together by the time they get to metaphase, and that makes it a certainty that one can go one way and one can go the other way. I can't screw this up. Okay, so there are some assurances here. But we can screw this up.
Oh, well.
What would happen if both members of the pair accidentally went into one daughter cell. What happens if this microtubule attaches here, but for some reason this microtubule from the spindle apparatus hasn't attached yet by the time the nucleus gets this, or not the nucleus, but the chromosomes get the signal to start separating? Remember, we said there was that whole really, really complicated process where these things send out signals saying, we're not ready yet, we're not ready yet, we're not ready yet, until the microtubule actually attaches. Then they stop sending out that signal. And when all of them stop sending out that signal, then things happen. Well, what if this one wasn't actually attached yet and all four of these go one way? What would the result of that be? I have 4 total cells that come out of this process, right? 2 of those cells are going to have what, and 2 of the other cells are going to have what? How many chromosomes is this daughter cell going to get? If the one that's not attached, if they all 4 go this way, how many chromosomes is this other cell going to get? 0, right? Okay, and so when that daughter cell also divides in the second meiotic division, if these are sperm cells, I'm going to have 2 sperm cells that don't have any of that chromosome. Okay, I'm going to have 2 sperm cells that don't have any of that chromosome. And I'm going to have 2 sperm cells that have 2 of that chromosome. I'm only supposed to have 1 of that chromosome as a sperm cell, right? I only have a haploid set of chromosomes as a gamete. But if we screw up here and all 4 of these go into that first daughter cell, and then that daughter cell divides again, we're going to have diploid chromosome, whatever this is. And so what I've said here, what I'd like you to write down is 2 of the final gametes would be diploid for that chromosome number, while 2 of the final gametes would be aneuploid. And let me spell that for you.
Is that definition we need to know?
Yeah, aneuploid. And it's just like putting A with some other thing, right? Biotic or abiotic, right? This is like adding a negative in front of ploidy. So aneuploid, A-N-E-U, aneuploid means no chromosomes. Haploid means 1 set, diploid means 2 sets, triploid means 3 sets, tetraploid means 4 sets, aneuploid means no. So if this was chromosome 17, then 2 of the cells would be diploid for that chromosome, and 2 of the final cells would be aneuploid. Okay? Either is going to be bad. If I had a sperm cell that fertilized an egg that had no chromosome 17 in it, that would be fatal. Yeah. A-N-E-U. A-N-E-U ploid. Now, did we not have time, or did I? Or did all of you get that?
Yeah.
Sorry. I got anxious to the moment.
What's the last word?
Aneuploid, A-N-E-U, P-L-O-I-D. Uh-huh. Okay, now here's my question For some reason, I think about things like this. And what I'm thinking is, well, why can't the cell do this? This is what happens in mitosis, right? The individual paired chromosomes line up on the metaphase plate, not the homologous pairs. The individual pairs line up. But if I had a homologous pair where, and remember, all four of these chromosomes are now mixed, right? They're not either really mom or dad, although they originally came from mom and they originally came from dad. Neither one is really mom or dad. They're all four unique mixtures. Why can't I do just what I did in mitosis and have these individual chromosomes line up singly like this? Here's mom's duplicated chromosome number 1, again, though it's all mixed, and this is dad's duplicated chromosome number 1. Wouldn't that result in the same thing? Wouldn't I still get 2 chromosome 1s in one daughter cell and 2 chromosome 1s in the other daughter cell, just like I would if I did this and I did this? Wouldn't I still get the same result? I think I would. I'd still get diploid daughter cells in the first meiotic division. I would have 2 chromosome 1s in one daughter cell and 2 chromosome 1s in another daughter cell, 2 chromosome 2s in one daughter cell, 2 chromosome 2s in the other daughter cell. Why can't I just do that and then my daughter cell would have a chromosome 1 and another chromosome 1, and my other daughter cell would have a chromosome 1 and a chromosome 1. And then I could separate those 2 cells and have a chromosome 1 here and a chromosome 1 here and a chromosome 1 there and a chromosome 1 there. Why can't I do that? What about this cell makes it so that would be a real problem? If I just had a bunch of individual chromosomes in this first meiotic division, after that, what would be the problem with that? Any ideas? How would I make sure that my next daughter cell got an equal distribution of all the chromosomes, right? Because it's only when they're in the paired state at metaphase that I can ensure one daughter cell gets one of each, right? If they're like this, if they're individual like this, then I can't be assured that this cell won't still get both chromosome 1s and this cell won't get any chromosome 1s, right? Because they're not attached. And it's only if they're attached during the second meiotic division that I can make sure that each subsequent cell gets one. So what I said, why can't the individual duplicated chromosomes, sorry about that, line up at the metaphase plate just as in mitosis? And so what I said, the resulting daughter cells from prophase 1 would still have the same chromosome profile as if homologous pairs separated. Let's say that again. The resulting daughter cells from prophase 1 would still have the same chromosome profile as if homologous pairs separated, but each of the chromosomes would be individual in this case instead of paired. There would be no way to reduce the chromosome number to haploid with a second cell division. Okay, and that's what I want you to write down. And I want you to try to understand why that would be the case. And answering these questions, if you understand the answers to these questions, you have a pretty good understanding of meiosis. Yeah, I'm sorry. I know.
It's too long and I cannot see.
And you don't want me to have to keep repeating it, right?
After we write it down, could you simplify it?
In what way? You seem to have an ability to say things more concisely than I do, so if you can come up with a better way.
Marshall, what is the character of the chromosome void?
Would be individual in this case instead of paired. I should have just typed this one out, huh?
Possibly.
Yeah.
What's the last sentence?
It says, there would be no way to reduce the chromosome number to haploid with a second cell division. Okay? If I've got individual chromosomes, I can't actually divide those, right? I have to have something that's paired to line up at the metaphase plate. Okay? In order for me to separate those cells, I have to have chromosomes that are paired. And it's only if homologous pairs line up the first time that can happen. The resulting daughter cells from prophase 1 would still have the same chromosome profile as if homologous pairs separated.
I said it would be individual.
But each of the chromosomes would be individual in this case instead of paired. There would be no way to reduce the chromosome number to haploid with a second cell division. This is hard, but it's so critical that you understand this. And hopefully it kind of brings about a sense of awe at how amazing this process is. We are all wonderfully made.
Surprising we exist the way we do.
As many things as can go wrong with this process, it is surprising. Are we all good with that then?
I think so.
Very little more writing here.
Maybe a few drawing.
Okay, now, the pairs of homologous chromosomes lined up at the metaphase plate separate. This phase is similar to anaphase of mitosis, except that instead of individual chromosomes moving into daughter cells, each pair, right? In anaphase of mitosis, I just have pairs of chromosomes splitting up like this. But in anaphase of meiosis, I have homologous pairs splitting up, right? Okay, so if you kind of keep that in mind, in mitosis, this is what happens at anaphase. In meiosis 1, this is what happens. So our goals are to separate the pair of homologous chromosomes into two daughter cells. And what we'll see is genetically mixed pairs of homologous chromosomes being separated, each pair will move to a daughter cell. And as with mitosis, in the cytoplasm, we'll see the spindle fibers shortening and the cytoplasm elongating. So this kind of illustrates that same thing. And is yours all blacked out? I'm sorry.
Yeah.
It's okay. My drawings don't really add that much to it anyway. So same thing after the pairs of homologous chromosomes separate into the daughter cells. The features of telophase and cytokinesis are similar to what they are except in mitosis, except that 23 pairs of chromosomes are still attached rather than separate. The nucleus reforms, the cytoplasm divides, but only for a short period of time, because then we get meiosis 2, the second meiosis. So for telophase and cytokinesis, we reform the nucleus, we disperse the chromatin, we break down the spindle apparatus, the nuclear membrane reforms, chromatin disperses, the nucleolus reforms, and don't worry, again, what I'll ask you to do as far as testing on this kind of thing goes is I will describe what the features are and ask you to tell me what phase that represents, okay? I'm not going to ask you to spit out all of the features of these things, although I would like to, but that's just unreasonable. So in meiosis 2, the second cell division, in prophase 2, chromosomes recondense, the nucleus breaks down, the spindle forms just as in mitosis. However, no genetic manipulation occurs. In metaphase 2, the 23 paired chromosomes again line up at the metaphase plate, and these separate in anaphase 2. However, this time there are only 23 pairs of chromosomes. Each cell only gets one. Okay, so now I have 23 pairs of chromosomes, and each daughter cell gets one chromosome. Okay, one of the 23. I am now haploid, right? Each of the 4 daughter cells that result are now haploid. So again, it all comes down to what are the goals of meiosis? I need to shuffle the genetic material. That happens in prophase 1. I need to reduce my chromosome number to haploid. That happens in the second meiotic division, right? I get that separation of the final sets of chromosomes, and now each daughter cell just has one set of 23 chromosomes and is now haploid rather than diploid. This process occurs where in the female? Where do I get this change from these genetically identical diploid sex cells to these haploid gametes? Where does that occur in the female?
The ovaries.
There you go. It goes in the ovaries. Okay? And produces 4 genetically distinct haploid ova for each cell. It occurs in the, where in the male?
Testicles.
In the testis, or the testes in the male, and produces 4 genetically distinct haploid sperm cells for each cell that begins the process. Okay? Does eat. Okay, so our goal for the next, what do I want to do? There's no way we're going to make it through all of genetics before break. So what I'm probably going to do is give you Next Tuesday, a study guide for a test over what we've covered so far related to cell division, and the rest of the time we'll focus on anatomy. Okay. And then we'll finish up genetics once we get back from break. Yay. 2 more weeks.